afternoon and a very warm welcome. My name is Rachel Bear and I'm the Dean of the Faculty of Natural, Mathematical and Engineering Sciences, and it's a real delight and privilege to welcome you all this evening to the last of this year's Inaugural Lecture Series, the Chemistry Inaugural Lectures. So these events celebrate wonderful professors who've either been promoted recently or within the last few years or have joined King's as professors. And it's a really great opportunity for us to learn a little bit more about their research and their journey to get to where they are now. So the first speaker tonight is Professor Andre Cobb, who was quite recently promoted to professor. A little bit of a backstory, although I'm sure he'll tell you more in the talk. He's one of our graduates. He studied chemistry at King's, including spending a year in industry at ICI, where apparently he watched paint dry for the year. Then undertook PhD work at UCL, developing new ligands for asymmetric organozinc and organonickel chemistry. After that, he went to do postdoc work at the University of Cambridge with Professor Florian Hohlfelder in biochemistry, working on synthesis and kinetic studies of synthetic enzymes. Then for Professor Stephen Lay in the Department of Chemistry, where he worked on the synthesis of anti-amyloid compounds and then the development of asymmetric organocatalysts. And that last field is where he contributed most. So in my non-chemistry language, I would say this is about creating clever and very sophisticated chemical molecules to do cool stuff in medicine. Is that a reasonable? It'll do for now. You'll hear more when he explains it. So he started his independent research career at the University of Reading and then returned to us in 2016. And we're really delighted to have Andre as part of the faculty and part of the department. He's a real team player, and even though he's a supporter of West Ham, he is a real good team player in the department and in the faculty and the university. I was just talking to my boss, the senior vice principal, recently, and she was mentioning, oh, Andre, he's been a great member of academic board. So, really appreciated all your contributions as well as in chemistry, in the education, in the research, and more general service. So thank you very much, and we're delighted to learn more about your research.
This is on now, is it? Okay, fantastic. I don't have to stand near to this speaker. Right, thank you, Rachel. Thank you, Carl, and the organisers for making this happen. It's a real privilege to be stood in front of you today. So this lecture is a bit unusual for me. I'm not normally talking about myself. Normally it's quite a technical talk that I would give, and this lecture is not going to be technical at all, maybe a little bit, but not too much. So it's about my career, and so I need to push that on. So the title of my talk is The Total Synthesis of an Organic Chemist. So a total synthesis, for those who aren't chemists, is the phrase that we use to describe the synthesis of something very complex from very simple things. And this is a case in point here, this kind of helix, which actually I generated from AI. I think it's quite impressive. I didn't actually build that myself. So this is a relatively complex structure, this helix, that is comprised of very simple components. And there I am stood by it. So you could argue that an alternative title for this talk would be How I Never Really Stopped Playing with Lego. And I'm sorry if this gives us some problems on YouTube and a cease and desist from Lego, but I'm going to unashamedly use the term, I'm afraid. A total synthesis has very many different steps. The shorter the total synthesis, the better. My total synthesis is quite long, actually. It took me a long time to get here, starting as an undergraduate at King's here in 1993, so 21 years ago. But originally it started with a desire to design and build. That cute kid there is me, same haircut and everything. This is probably the age I started trying to play with Lego and building more complex things from more simple things. I don't have a picture of myself doing that, but I do have one of my daughter. Here she is, and she's building a helix as well. I would call this an I to I plus 22 helix, which means that if you look down the barrel of the helix, that one complete turn of the helix has 22 units within it. My daughter's 18 in a couple of weeks, so she's grown up a bit, and sadly she wants to be an engineer. Actually, I'm very proud of her. She wants to be an engineer, but she's not a chemist, but I'm still proud of her. So if I were to ask you, what is the best Lego set in the universe? It's not the Lego Millennium Falcon, and by the way, it's almost Christmas. It is actually the periodic table. Of course it is. This is the best Lego set in the entire universe. I just put it there for your, to emphasize that. And I guess this is where I have to confess a little bit of envy of my inorganic colleagues who get to legitimately play with the entire Lego set. But that's okay. As an organic chemist, I get to play with the best Lego brick of all, which is carbon. Carbon, the basis of all life, of course, on Earth and probably in the universe. I think my Origins of Life colleagues would agree. Because it can link to itself in so many different ways and produce so many different structures. So I've got some examples of that up here. These are just randomly selected. But this shows you the great diversity of structures that you can have with carbon. So those of you who aren't chemists might wonder where all those C's are, but we do this shorthand thing where we just draw the skeleton of the structure. And at all these intersections is essentially a C. So we can start with relatively simple compounds like amino acids. And I say relatively simple. When you start linking these together, you get way more complex structures, and I'll kind of touch on that later. One of the loves of my life, sugar, of course, is a beautiful disaccharide there. You've got structures like strychnine, look at that dense functionality. And I should say that this is the kind of thing that gets an organic chemist going. Yeah, I say that to you emphatically. It's basically this idea that we can try and build complexity, and we want to do that from simplicity. That's a term I'm going to use quite a lot, complexity from simplicity. So designing chemical reactions that can achieve this kind of framework. Of course, nature does it with ease, nature being the best synthetic chemist, but we want to be able to do it in the lab. So strychnine, of course, is beloved by authors like Agatha Christie in various books. Glyceric acid, does anyone know what that is? I'm quite happy for an interaction here. We don't have to just listen to me. Put your hand up if you know what glyceric acid is. It is the the sweetener component in licorice. So whether you like it or hate it, if you hate it, it's probably because of its compound. If you like it, it's probably because of that compound. Also, I understand it does something strange to your potassium channel, so don't eat too much of it, because I think there's a case where it was fatal back in the day. Talking of fatal compounds, this is a ciguatoxin, which is a marine toxin. And look at that beautiful structure. All these snaking of heterocyclic rings to the fangs of a dial is not a compound you really want to meet. But do you know where you would see it? I've already said it's a marine toxin. And it's found in the coral reef fish. So Nemo here isn't maybe as friendly as he looks. But to be fair to him, I don't want to introduce his name. He actually isn't the origin of this toxin. It's the algae that penetrate his flesh that produce this. So there's one other thing I wanted to mention about these, for those if you don't really do much chemistry, and that is these weird bond projections from those carbon atoms. So you've got this wedge here, and you've got this dash here. So what do they actually mean? Well, I might just go on to the next slide to show that. So here we have that basic amino acid I showed you. This is alanine, and one of the bonds is pointing towards us. That's what this wedge means. And this dash means that this substituent is pointing away from us. And this is by virtue of the fact that carbon can have 4 bonds attached to it. So what's not shown here is the hydrogen, same here. We're trying to do as much shorthand as we can to stop confusing things. But you can see that if we were to rotate this by 180 degrees, so the nitrogen is now on this side, that we would get, in 3 dimensions, this structure, these 2 structures here. And you can see instantly that they are mirror images of each other. Not only are they mirror images of each other, they are non-superimposable mirror images of each other. And something that's cited from school upwards is like the left and right hands are mirror images and you can't superimpose them. So the thing about these mirror images is that they will interact with biological systems in different ways. I'm not going to the how and why of that, but essentially there are lots of quite nasty examples of where this is a bad thing. This is meant to be a happy lecture, right? So I'll stick with a more benign example of limonene, where you can see that one mirror image has this effect of smelling like lemon, pine, and turpentine. You can't really see the eye there, can you? But this other mirror image is something that smells like oranges, and that's just because they interact with your nasal receptors in different ways. So that's the effect of mirror images. And the only reason I mention this along the way is that actually this has been the major part of my research is how we control the formation of one hand over the other. So can we produce exclusively one mirror image? And this is a field called asymmetric synthesis that I'll talk about later on when I get to my career. Right, so step 2 in the total synthesis of an organic chemist is to study chemistry at a very good university. And also I mental note to say all natural sciences, which we now offer everyone who's on YouTube, because you've got to do the pitch as well. So chemistry or natural sciences at a very good university, and some of you might have come through these doors just outside there. And this is the class of 1997. Oh, I feel old. And there's me sat there looking as smart as ever with the same haircut again, basically. And this is the other view here, which is of the Strand building. This is where the chemistry department used to be, at the very top floor of there. What's at the top floor now on the 8th floor? Is it the English department or something? Is it? I don't know. I don't think it's an NMES, but anyway, it had a really great view of the Thames. I do miss it. But yeah, so this was the first place I got to do chemistry research, of course, got a flavour of trying to do discovery reactions. And that was, of course, with my final year project. So when I selected my final year project, I wanted to do organic. I was very clear from the beginning, I wanted to do organic chemistry. But they gave me an electrochemical project with Martin Boutel, who was an electrochemist at the time. And I complained bitterly. This letter didn't come by email, because the email hadn't really taken off by then. And I got a letter through the post at home and instantly went straight back into King's and met the head of department, Frank Hibbert, and complained bitterly to him. I want to do organic chemistry, I don't want to do electrochemistry, although I appreciate the origins of electrochemistry at this college. And he said eventually that I'll be working with Kamiyar Afarinkia, who was a new organic chemist at the time. And he reasoned that the reason he didn't allow me to, or initially didn't give me an organic project, because I might get favoritism from the NMR spectroscopist at the time, who was my dad. Clearly you didn't know my dad, but you might recognize the Strand brickwork there when it was in that building. So yeah, I went ahead and worked for Kamiyar. I'm not going to go through the chemistry, because I promised I'm not going to be too technical here, but essentially the project I did, it was a nice little short project, and that was a synthesis of a beta-phospholactam, or an azophosphetidine. And the reason Kamiyar was interested in this structure was because it's quite a mimic of a beta-lactam ring. And we all know the oncoming storm, we should say, with antimicrobial resistance and the need for new antibiotics. I'm afraid I don't know where that project went, actually. Probably I should find out. So the next step is to do a PhD in something that you love. And I've already mentioned asymmetric synthesis as being love of my chemistry undergraduate days. And so I went to UCL. Actually, I probably shouldn't say, of the 4 of us, 3 of us went to do PhDs at UCL. I'm afraid we were a bit turncoaty. But it was a great time. We stayed in London, and there's me. And if there are any safety officers in here, try and spot the 2 deliberate errors in my attire, which would be, well, okay, just look at Alf. He became a policeman, by the way. And he is obviously wearing a lab coat, and he's wearing his safety specs in the correct way. I've got my safety specs protecting what I think is the more important part of my head, the brain, and not wearing a lab coat. So a really good way of showing, well, my students are here. You don't do that, by the way. Don't do that. And I worked for this guy called Charles Marston, and I was doing asymmetric synthesis. Published a bunch of things during my PhD. Again, I'm not going to go into the technical aspects of this, but one thing I want to highlight is this figure, this 80% EE. So EE stands for enantiomeric excess, and it describes the predominance of one hand over the other. So 80% means you've got 90% of one enantiomer, one mirror image and 10% of the other mirror image. So 90 take away 10 is 80. So an enantiomeric excess in the 90s is generally quite good and quite selective. So after your PhD, it's the usual, I'd call it conveyor belt, but an important part of your training is to do a postdoc. And so what I decided to do, as Rachel mentioned, is do a postdoc in a biochemistry department, because I wanted to do something completely different. I wanted to do something biological. I wanted to cure cancer, right? Okay, I mean, I thought that was naively at the time something I could do. Turns out Florian really wasn't working on that kind of area, but what he was doing was catalysis. So catalysis wanted to stay with me, basically. This wasn't asymmetric catalysis. We weren't trying to develop one hand over another, but we were doing this thing called hydrogen bonding catalysis. And this is something, again, that reappears in my career later on. So it gave me a real appreciation for the power of hydrogen bonding catalysis. And it also gave me a really good training in kinetics as well, how to measure these rates of reaction, which is important for somebody working in catalysis, I think. It was only a short stint there, because after that, I went down Tennis Court Road, across Lensfield Road to the chemistry department to work for Steve Lay, who was basically one of the most prestigious groups in the world to work for. So I don't know how I managed to do it. I just got quite lucky. I was in the right place at the right time, I think. There I am here. This is actually his 60th birthday, which convened after I started my first academic position. But you can see this is just a selection of some of the people who went through his lab. And there are some real big names here that you would know in the organic chemistry community, including the current 1702 chair, Matt Gaunt. And that was Steve's chair at the time, 1702. So I went to see Steve a few weeks ago, and he's looking very healthy. He's still got a bigger office than I have. And he doesn't have to share either. So he wanted a photo in front of the very first 1702 professor at Cambridge. But it was really nice to see him. I hadn't seen him for a long time. Actually, the last time I saw him, at his 70th birthday symposium, he said to me, "You're doing really well at Reading, but you've got to publish more." So, thanks, Steve. I knew that. Right, okay. Staying with Cambridge, this is me around that time, partaking in the Cambridge lifestyle, shall we say. And published a whole bunch of stuff with Steve. And you might think, ooh, Nature paper, I'm going to talk about that. But no, I'm not going to talk about that. I'm going to go to the other end of the impact factor, to the Sinlet here, which for me was the most influential paper on my career. So what I was doing with Steve at the time was developing catalysis again, asymmetric catalysis. And we were making reactions that generated two stereocenters. In my PhD, you might have noticed I've only got compounds with one stereocenter. So now we're increasing the complexity from simplicity by making two stereocenters. And you can see the selectivity, that EE value I mentioned is really good here. Oh, and it wasn't all play, all work. It was some play as well. And this was a 70s party in the Whiffin lab at Cambridge, as it was called. And I think this is at the other King's College there. And there's me. I'd like to say I dressed up for the 70s party, but I think that was actually probably my normal attire. And you can tell it's me as well, because I'm not wearing safety specs or lab coats. So. Okay. Then this is the next step, and this is the one that really confuses me. I have no idea how I went from being a postdoc to getting an academic position, because today the competition is so furious. I just thank my lucky stars that I'm not in today's cohort of postdocs looking for an academic position, because I don't think I would stand a chance, if I'm honest. The talent out there is just phenomenal. But through luck or magic, I managed to get an academic position at the University of Reading. It was a school of pharmacy, so not the Department of Chemistry, which sadly has had a bit of a problem recently. And this is me, plucky me in 2005. And this is me 3 years later. Yes, looking worse for wear. That's what 3 years of academia does to you, by the way. Also the birth of a child, but that's, aside from that, it was a fun time building your group up in those first few years, and I published quite a few things in that time, and I'm just going to stop on this one and highlight that reaction. Again, I'm not going to go through any of the science, really, but to point out that we're making systems here now which have 3 stereocenters. I think I mentioned my PhD having 1, postdoc 2, and now we're getting even more complex. So 3 stereocenters here. And just from this really simple starting material. And we use another type of catalyst to do this, and you can see our selectivities were fantastic. And at the time, really, I was only thinking, I wasn't planning in advance or kind of reasoning why we were doing this, which I probably should have done. I just wanted to know whether we could get this functionality to link with this functionality. So we're just forming a new bond between this carbon atom here and this carbon atom here. If I thought about it, I would have kind of realized that these are gamma amino acid precursors. And gamma amino acids are used therapeutically as anticonvulsants. So gabapentin is a great example of this because it's quite similar to the structure that we've made. I know that may not look too similar, but you've got this ring system here, you've got a ring system here. This you can turn into an NH2, and this you can turn into an acid quite easily. So it's almost what we call a constitutional isomer of gabapentin. But I've never actually explored its anticonvulsant activity. And actually, being at King's, at the new professors meet, I met a guy from IOPPN, which is our basically they do physiology or something, who says he can test these compounds, so it's quite good to come back here and have that opportunity. It's quite old work now. So the next step in the total synthesis of a chemist, I'm sure all academics here will appreciate this slide here, the Just Keep Swimming for another Nemo reference, which is that above, whilst we're giving lovely presentations and we're writing lovely papers, giving good lectures to undergraduates, underneath we're swimming like mad to try and meet deadlines, try and get feedback going, and writing the grants that give us the money to come and do this kind of work. But then nevertheless, you just have to keep going. And this isn't just step 7, this is ongoing all the time. It's just part of the job, really. So we've done that, and we've kept going, and this is the group a few years later. Again, we published a whole bunch of stuff, and I'm just going to stop on one again, which kind of continues that previous story, where we've used a similar type of catalysis to generate systems that are even more complex, with up to 4 stereosyntheses now. So we've gone from 1 to 2 to 3 to 4, from relative simplicity. So these are quite simple compounds. You have to make this one, but you can buy this one. And we can put these together with a catalyst and get up to 4 stereosyntheses. Not only that, we can make 5 stereosyntheses using this methodology as well. It's, I don't know if we're trying to show off. In fact, so the yield isn't amazing. But again, the selectivity is very good. And this is a crystal structure to demonstrate that we've got those 5 stereosyntheses. So this leads me to step 8. I mean, you look at these compounds, you think, they're nice, they've got a cool number of stereosyntheses, but what use are they? You can't do anything with them. I mean, they're kind of gamma amino acid precursors, I guess, but it's a bit of a fudge because you've got that on there as well. So that leads me to step 8, which is that you've got to demonstrate the applications of your reaction. So I mentioned at the beginning I didn't give much thought to application. Subsequently, when I realized the funders do care about that kind of thing, I started thinking about application very much. And so one of those was to take that compound on the previous slide, and what Raj did was he converted it to this compound here, some really cool chemistry there I'm not going to go into. And this is a tetracyclic system, so it's 4 cycles fused together. There's a crystal structure. And this is good because it is like a natural product, like a rain, which you get from a yellow rain lily. And it's a great opportunity to show a picture of a nice flower on my slides, aside from all the dreary chemical structures. And it's probably hugely toxic as well, but Raj is still alive, so it's probably fine. Yes, so here's another example of application and also of generating molecular complexity from relative simplicity. So we have these 2 structures here. You can buy this one. Again, you can make this one easily. And just using a very, very simple catalyst, we can make this tricyclic species with 1, 2, 3, 4, 5 stereosyntheses. Oh, I didn't mention is that previous slide. Getting 5 contiguous, as we call them, 5 stereosyntheses from just 2 achiral starting materials was a record equaling attempt at the time. Hopefully this one will rotate. Yeah, so you can see that this is the system we've made, and this is the structure we obtained. This is the crystal structure of that system. And the reason we were interested in these was because we were targeting some steroids. And if I show you what those steroids are first. Yeah, so here are some of the steroids. These are called the cardiotonic steroids. They're hugely toxic and will cause you a heart arrhythmia if you have sufficient quantities of them. Of course, We all know foxgloves due to toxigenin, which you are told not to touch in the garden, at least wash your hands afterwards. But this structure is ubiquitous across many different species, including this fella who kind of reminds me of Baron von What's-His-Face from Danger Mouse. So what we wanted to be able to do was demonstrate that we can convert this structure here into this structure here. We wanted canogenial because it seemed the easiest and most appropriate of all those targets. This leads me to the next step, which is in the total synthesis of an organic chemist, which is to fail as well sometimes. We're not total synthesis chemists, really, but we tried to be. Steve Lay, who I was with, was a very good total synthesis person. So I figured, it can't be that difficult. And so Dan had a go. He did a really good effort. Sadly, we didn't get to our target for, again, chemistry reasons I'm not going to go into. But we did get a new structure, which is kind of a steroidal analog, which had never been reported before. So we published on that as well. I think if he'd had more time, we could definitely have solved that problem. But he got towards the end of his PhD. Oh, yes, and this is beginning to get into the King's era now. Okay, so it's all very well just doing asymmetric synthesis, making cool molecules with billions of stereocenters. But it doesn't necessarily scream fundable all the time. So sometimes you have to, put your hands up and say, okay, I'm going to try and find a more fundable area, something that will bring money in. And my side hustle was cannabis. I'm sure none of you recognize what that plant was. But so I wasn't the Walter White of Reading at the time. But what we were interested in was making, let me just push forward a couple of things. So what we were interested in was making metabolites of cannabidiol, which is a compound that you may have heard and you've seen on the high streets at questionable pharmacological quantities. But what GW, who are the company trying to market this, wanted to do was use CBD to treat a childhood epilepsy, an orphan disease known as Dravet syndrome. In order to get FDA approval, they had to demonstrate that they knew what the metabolites of this thing was, what the body converts these structures into. And they suspected some of these, and we synthesized them for them so that they could use them as markers to demonstrate that they knew what the metabolites were. It was very easy chemistry compared to the stuff I've been showing you. Actually, this one was a little bit tricky. But despite being quite easy chemistry, it's certainly the most impactful thing I've done in my career so far. Anyway, you can't keep a good synthetic methodologist down, can you? So we wanted to develop some asymmetric routes to CBD analogs. So this is the analog we went to make. You can see it's a bit like this one here, except we've just tethered that side chain up to here. And in doing so, we create a new stereocenter, and we wanted to be able to control that stereocenter using asymmetric catalysis, which we did, and we published that, and we patented it as well. So that was Laura who did that work. Okay, so almost at the end now. Have I been going for 49 minutes? I'm so sorry. I didn't realize I've been going that long. Oh, okay, sorry. Right, so I'll be very quick. So this is another area that we've recently become interested in, which is to develop methodology to these compounds here, which you could never get to before, and incorporate them into a linear molecule. Although it looks linear, and these methyl groups look far away from each other, it actually folds into a helix. Oh, already press that. So you can see from this movie that there is a hydrogen bond between here and here, and that's what lets it fold into a helical shape. I'm going to just crack on because we're on really, really, really close to the end. So it just comes back to playing with Lego, essentially. So we've made other helices in this way, and you can represent those in Lego terms. I had great fun with a package that does this. So these are the alpha amino acids that represent here, and we can intersperse it with other unusual amino acids, and we can represent those with that type of Lego system, and we can mix and match them to get this type of helical shape. And what's really good is that functionality can be brought quite close together, and you can exploit it. I'm just going to push forward to show you how we did that. So we developed a helix that was able to catalyze the degradation of this compound here into these two component parts. I promised Rivka I wouldn't filibuster her talk. I'm not doing quite so well, actually. Okay, and just finally, now, what about the future? So helices are, even the choir are trying to get me to stop. Helices themselves are also handed like the left and right hands. This is a picture I took in Sorrento earlier in the year, and you can see this is a right-handed helix, and its reflection is a left-handed helix, so they're non-superimposable mirror images. And the question we're asking now is, can we use that chirality, as we call it, to control the formation of left and right hands of other reactions? The other thing we want to do is be able to build a helix first, and then attach functionality to it afterwards using something called click chemistry. So in Lego terms, you can see that will only combine with this one, and this will only combine with this one, and not the other way, which means we get lots of control over the functionality and its placement. And also, whilst we're trying to keep our hand in novel synthetic methodology, and then step 0 to infinity, which I missed out, surround yourself with awesome people, because none of this would have been done without them. I didn't do any of the work other than the PhD and postdoc, and all my collaborators as well. And this is my group over the years, stopping at a trip to Legoland for obvious reasons. This is us bringing us up to date now, and this is about a month ago, this last one, just near Southwark Cathedral. I want to thank my funders, and I want to thank my family, some of whom are here, my mum and dad, and my wife, and that's my sister up there, my kids. So thank you very much, and I'm so sorry I ate into your time. I had no notice of that. Thank you.
Hi, so I'm Paula Booth. I'm from the chemistry department. I had the pleasure of recruiting Andre back to King's. Oh, we've got a rush of sitting down. There's plenty of seats at the front. Oh, someone sat in my seat. No, it's okay. So I have the honour of doing the vote of thanks. So a little bit of context, some things Andre didn't mention, but at times, being Andre, he gets very enthusiastic, so I might have missed it. So as he said, he was an undergrad here, but King's, in their great wisdom, alongside some other departments in the country, at one point made the quite catastrophic error, let's face it, of closing chemistry completely. Nothing to do with Andre, although it did happen shortly after he was here, without the safety specs, so who knows, quite frankly. And so we had to completely restart. And so this was, quite a thing. We had to start from scratch. None of that distant view of the labs up on the Strand somewhere was still in existence. So there was nothing for chemistry. And we had to recruit people who had to join us in this endeavor and believe that, things were going to happen. And Andre was great, actually, because he was, I have to say, ridiculously keen to come back to King's. He was so keen that I possibly took advantage of him, poor guy. So he turned up. The good thing was he actually knew something about the old department because no one else spoke about it. It was like, oh, did we have a chemistry department? No, I didn't close it. But Andre actually was really, he spoke really well of the old department, and he was very, very, pleased to come back and get chemistry going again. And he ended up doing a lot of teaching, which he continually reminded me of, I do remember. This was because we were pretty short-staffed at the time, and we had very few of these people who play around with carbon, and it's quite a big thing. They do like to teach people a lot about organic chemistry, organic chemists, so we had to do a lot of teaching. He also was then, constantly saying yes to me. He was doing exams. He was then doing safety, and before he knew it, he was like representing us at faculty level. Nobody ever really wants that job. I don't know how we really ended up there. And looking back, maybe it was a mistake, actually, seeing that no lab coat, no safety specs business, Andre. So maybe, yeah. I'm also wondering why the little Lego guy at the beginning didn't have the specs on the head. But this might be because he's now a professor, so he sits in front of 2 screens in his office looking out the window and doesn't even have safety specs on his head. But anyway, Andre has flourished here, and I think the thing about him is not just his enthusiasm, but he's just such a nice person. I mean, as I said, he was always agreeing to do things. He helped us out of all sorts of binds. I remember we had a member of department leave suddenly, and Andre was then filling in and going out of his way. And he's the sort of person who just made the department work. And he's just such a nice person, you know? You're always waiting to find out what's wrong with him. Why is he so nice? Are we one day going to find out? But he isn't. He's just lovely, and he's so popular. And I'm really, really pleased that he got promoted to professor at King's, because I know how much he just really likes being here. And so if you can just join me with thanking Andre for such a great talk. And for sticking with us.
Thanks, Paula, and thanks, Andre. And it's not just one talk, we have two talks. So Rivka, Professor Rivka Isaacson, Professor, when did you become a professor? It's a few years. Yeah, a couple of years ago. So a bit of back story here. Not one of ours, but not too far away. So studied biochemistry at the University of Manchester, and then chemistry in Cambridge, doing a PhD there, and the supervision of Alan Furst. Then went overseas to Harvard Medical School to do postdoc work with Professor Pamela Silver, and then back to Imperial with Professor Steve Matthews. So I don't know, when you two interacted at any of those places in Cambridge or Imperial, we'll pick up that later. Yeah. Now, one of the things I've had privilege to see in action since joining King's is Rivka's passion for interdisciplinarity. And we're not just talking about chemists talking to biologists here, but working in collaboration with fine art. And I also had reference to the Iris Murdoch Research Centre at Chichester. So lots of really exciting collaborations, really wide-ranging. In terms of service, Rivka serves on several national committees and steering groups, UKI Physics of Life, work in this NMR space, and has a couple of prizes to be mentioned. So winning the Judith Howard Prize from the Biophysical Science Institute, and the 2024 winner of the British Biophysical Society Elspeth Garman Prize. I do like the tagline of professional scientist and amateur artsy person, which appears on the Instagram for Rivka. And I've really valued her really dedicated work as director of our graduate school and the faculty. And I know she takes particularly the sort of pastoral care and the support for our PhD students, both in the department and more widely, really, really seriously. So thank you for all of that, and we look forward to talk number 2.
Deep breath. Hello, everyone. It's amazing to see you all here, and a shout out to the people on the live stream as well. I know my parents are there. And a special thanks to my sister Dina, who came all the way from Manchester, and my brother Shlomi, who came all the way from Gateshead. And whenever I say my sister, everyone knows this. If I say my sister without qualifying, I mean Shan, who's come all the way from Archway. Anyway, I first want to explain my title. So I had a teacher in primary school who told us that in order to maintain your religious observance, it's like walking upwards on a down escalator. So if you don't make any efforts at all, you're just going to go down. And even to stay in the same place, you have to make a lot of effort. And then so to get anywhere in terms of improvement, you really, really have to make an effort. And although that is somewhat of a stressful concept, I have found it applies to so many things in my life, all the way through, I mean, since I was 10 years old. And actually, it's quite a useful way of explaining proteostasis. So this is an umbrella term under which all of my research can fit. And to explain it, I just first wanted to ask you to imagine your body divided into 30 trillion separate compartments. And the reason I'm asking you to do that is because it really actually is divided into 30 trillion separate compartments. And each one of those is really, really small, as you can imagine it has to be. And each one is really, really crowded, and there's tons of stuff going on. You can kind of see it a little bit in this, this iconic image from David Goodsell, who's one of my heroes because of being a scientist and an artist, and once reviewed one of our papers. Anyway, yeah, so there's a lot of crowded stuff going on, and it's really important that certain components are present at one time, absent at another, that we have good spatial and temporal control of machinery in a really small subsection of our bodies. And so in order to maintain that, we have a lot of machinery, and that's what I'm calling proteostatic paraphernalia. And that whole process of keeping everything exactly the same or responding to different changes that need to happen is kind of like that walking up the down escalator, because a lot of processes have to happen in order for even the status quo to be maintained. I wanted to give I have many friends here who are not scientists, and also many friends who are scientists of different kinds. So I thought I'd start with a very brief science lesson, which is also what I teach my second year at Chemist as well. So there are three main biological polymers, as we call them. You have your DNA, which is hidden inside the nucleus of all of those tiny cells. The nucleus is an even smaller compartment. You can imagine we're really on a small scale. But actually inside every nucleus, in those 30 trillion cells, is 2 meters worth of DNA that's wound up very small. And that includes all the instructions for everything that needs to be made inside your cells. And because we want to keep the DNA perfect, like we don't want it to get any mistakes or mutations, which would cause diseases potentially, if we want to use any of the DNA to actually make something, it's almost like you've got this kind of inherited grandma's recipe book that's falling apart, and you want to maintain all those family recipes, but you don't want to bring the book out and do the cooking right in front of it and get it messy. So you take a little picture with your phone, and then you use that for your actual cooking. And that photo equivalent in our cells is called messenger RNA. And that is the sort of temporary copy of your gene that you're going to use to encode a protein. And everything I've done in my career has been about proteins. So for DNA, it's a really, this iconic double helical structure that you see in sculptures all over the place, as I indicate in my photo. But then that is copied into a single-stranded messenger RNA, which is used to thread different choices from a selection of 20 beads called amino acids that actually Andre very kindly introduced in his talk, onto a sort of linear, almost like threading beads onto a necklace, but then that linear shape has to fold into a very complex mechanical machine that has moving parts, and there are tons of different things like that inside our cells. And so the next thing I'm going to show, I really hope this works, is a movie, and this was made by Mickey, my friend who's actually here in the audience, and this is showing you the messenger RNA being used by the ribosome to read each codon, each bit of the gene, and work out which bead should be threaded onto the necklace. And then as it comes out, it starts to develop some shape, which is essential for the job that it's going to be doing. So this is something I like to show. I have to say it's a really ancient and very basic animation that I created before I knew Mickey, I think. Anyway, so this is a gene, a fictional gene, and it's just to show you how the DNA would be kind of related to the final protein sequence. So here we have a codon, so every 3 letters in a gene is equivalent to 1 amino acid. This one is called tryptophan. Then we have, I don't know why it's not working. Then we have an arginine. These are all the beads, which are the different amino acids. We have an isoleucine, we have a threonine, glutamate, and then finally another arginine. And as you can see, like in a linear way, they're really not much use to us. But I hope I can do this successfully. When they're folded into their final format, then they can actually form some kind of functional unit. And so it's been a huge mystery, for years. How does the linear sequence of a protein relate to its final fully folded form? And so now people probably have heard that the Nobel Prize in Chemistry was awarded this year to AlphaFold and also to protein design. AlphaFold is a way of predicting what the final shape's going to look like just from looking at the linear sequence. But the way that AlphaFold was trained and how it started in the beginning was by looking at lots and lots of experimentally solved structures that were done by lots of people in my community. So how do you solve the structure of a protein? There are quite a few different ways to do it. And I've been involved in most of them over the years. My first love is NMR, which is shown in the middle here. And this uses a very powerful magnet. It's a technique, and the other two techniques are X-ray crystallography and electron microscopy. There are also various other ways you can do it nowadays. But with NMR, it sort of fell out of favor, partly because it is quite laborious. But I think in the era of AlphaFold, where a lot of structures can be predicted, it's actually really going to come back into its own, partly because you can do relatively simple experiments to validate predicted structures and check whether they really are true, but also because NMR can look at motion, movements of molecules, and that's really the next frontier of stuff that we need to understand about all this machinery that is inside our cells. So this is me holding a 3D printed ribosome in the protein data bank in Europe. This is probably 50 million times bigger than a real ribosome, which is the machine you saw earlier in the snowy video made by Mickey. And this was pretty amazing. You can take it apart. It's held together with magnets, and you can see the messenger RNA and all the individual amino acids that are being delivered to expand the chain. This was created by an American guy called Mark Holzer, who has an amazing website full of exciting things. This structure is based on the Nobel Prize winning structure of the ribosome that was awarded in 2009. And it was solved by Venki Ramakrishnan and Tom Stites and Ada Yonat. And I really enjoyed playing with this machine and gave it back and then discovered that it cost $5,000. I was quite relieved that I hadn't known when I was actually holding it. But actually, years later, when we had our Viewing the Invisible exhibition, my artist friends, some of whom are here, said, "You've got to get that sculpture, you've got to get it." So I wrote to the guy and I said, "Oh, do you know what, can I borrow it?" And he said, "Oh, I took it back to the US, but he's just making one for Venki Ramakrishnan to put in the lobby of the LMB in Cambridge. So just write to him and ask if you can borrow it before." And I wrote to him, and he said, "Sure." So anyway, the lesson I take from this is if you don't ask, you don't get. And that really is a message of my life. Like, just be brave and ask for things, because that was very cool. Anyway, to talk about my career, I don't actually have a timepiece, which is bad, so I don't know how long I've been going. Oh dear. Anyway, someone just yell when I have to stop. So the first place I was at in my education was Manchester University, which has already been mentioned. I did a year in industry. Well, it was called that, but it was actually in an academic lab in the US. This was my mentor, Professor Darwin Prokop. Amazing, really amazing guy. He actually only just died earlier this year. He was born in 1929, and he'd kept his lab going until just before the pandemic, which is just crazy to think about. They do that sort of thing in America, like just work for so many years. But he was a very nice and great mentor. And being in this lab was really what gave me the idea that I will be a scientist. I hadn't thought of it before that. Yeah, and we published this paper. It's still my, by way far, most cited paper. And in contrast to the model of retirement, I wanted to mention Steve High in the picture above. He's a long-time collaborator, but he was actually my lecturer when I was an undergrad in Manchester. And he was also lecturer of my PhD student, Beth, who's in that photo many years later. And he has really embraced retirement, having retrained as a yoga teacher, become vegan, and spent a lot of time in the Lake District. So just to show you a few models of how this stuff can be done. Anyway, next up, I was in Cambridge, where I met lots of my amazing friends who are here today, actually. And yeah, I really had trouble finding my photos from the lab. I don't know where they all are. So what I've shown as my photo is, this one really is historic, from a lab Christmas party. I worked on quite an obscure disease, as you might be able to tell from that title. But this paper really opened, I can't even tell you, opened a lot of doors for me. And I think I didn't quite understand what that was doing for my career, and it probably, in a way, it was a sort of disadvantage. Like I say that in lessons to my former self, that if I'd been a bit more conscious, yeah, I would have understood efforts that I needed to make later. One thing I was thinking, listening to Andre's, is that my career, I think, has very much not been by design. It was much more by accident. Anyway, so I was also wanted to show a picture of, I recently went to Cambridge with my daughter. This was for the memorial service of Olga Kennard, an amazing mentor who, actually it was her idea that archiving molecular structures, I mean, among other people. And so she started the CCDC, which was the forerunner to the Protein Data Bank that I mentioned before, that was used to train AlphaFold. So the impacts of these people is actually very long-lasting. And when I went to Olga's memorial service, I went for lunch with Alan First, my PhD supervisor, who's just had a knee operation yesterday, so he couldn't come. But he has not changed. He looks exactly the same. And that's his lovely wife, Marilyn, as well. Okay, next up, I went to Boston, to Harvard, which was also a very exciting experience. I had a fellowship from the Wellcome Trust, the only time I've ever had a lot of money from them. But that all might be about to change, who knows? Anyway, yeah, so my supervisor was Pam Silver, and these are some of the people from the lab. And the main, I just wanted to mention this T-shirt. This is the back of the T-shirt we all got from the Harvard Structural Biology Retreat in 2001. And the thing that's interesting about it is that, you know, it's the Harvard coat of arms with the three types of techniques very siloed, whereas now, if you're worth your salt, you basically have to do all of them. But that's a real example of kind of interdisciplinarity, which is my whole way of operating in life, and it's very important to me as a way of thinking, like really getting input from lots of different people with lots of different ways of thinking. And I must mention Amy, who's in this picture at the bottom, because she's now a very long-term collaborator. We both have our own research groups, and we speak every Tuesday, and we've had loads of grants and publications together, and and I met her when I was in this lab, so it was a very important time for me in that respect. And these are my roommates who I live with, who, again, still in touch with all these people, and it's lovely. Next, I went to Imperial. So the Wellcome Trust thing had 1 year where you had to come back to England. That was a condition. And I wrote randomly to Steve Matthews because I knew he did NMR and I wanted to continue doing some NMR. And yeah, he just wrote back straight away, was like, sure, come. I don't think he realized I would be kicking around for like 10 years. Anyway, these were very, very happy times. This picture is from our first trip to CCPN in 2003. This is an NML conference that I've been to every single year since, apart from 1 year when I gave birth to Netta, and then I regretted not going so much that when I gave birth to Saul, I brought him as a tiny little baby with me, and he got a medal for being the youngest participant in CCPN. And actually, really recently, a random person wrote to me and said, you won't know who I am, but it was so inspiring that you took your baby to a conference. I've just had my first baby, and I took her to a conference. So you just don't know what impact these things are going to have. Anyway, this paper, I'm sure I'm just like a million years over time by now. But this paper, yeah, also I think probably helped me to get the job in chemistry department at King's, because let's face it, I'm not like a real chemist. But it's in JACS, so that's a chemistry journal, right? Yeah. Anyway, I've been here, so I got a fellowship finally after the years of trying at Imperial, and once I got it, I just thought, okay, now I've got my own money, this is the time to jump ship. Because in Imperial, they were not renowned for keeping on every random fellow that was, you know, in the department. So I started applying for other jobs, and I saw that they were reopening the chemistry department at King's. And yeah, these are just, I've got so many other people I want to like show, so I'm going to race through. But these are people from my lab over the years, and also my lovely lunch colleagues at the top. You might spot a familiar face in the middle up there. And yeah, I sort of pride myself on having happy labs, and I've had some really fantastic people come through over the years. This is the current complement. I think most, or if not all, are here. We're a girl power lab at the moment, not by design, but we're going to enjoy it while it lasts. I thought I should show like a tiny bit of science, perhaps. This is what we fondly call the dancing chicken. This was made by Sasha in our lab, and it's what I was talking about, motions. It's a cryo-electron microscopy structure that is still in progress, but it's part of the degradation machinery. Like when I talked about proteostasis, you have to have ways of taking things out that, you know, are finished with or obsolete or causing problems. And part of the degradation machinery is this round thing. You can see in the middle there, like something that's being unfolded by a protein that's being sent from its three-dimensional shape back to its linear sequence to be chopped up and used, its amino acids reused. Yeah, I don't know if anyone can tell me like how long I've got. But yeah, anyway, I just wanted to run through the other things. My eyesight is so bad that I can't read it. 3 minutes, okay. Yeah, so I've done a lot of stuff, and I just wanted to give a shout out to the people who believed in me, particularly Katie Guest, who commissioned me to review an A.S. Byatt novel in The Independent on Sunday, literally without knowing whether I was capable of writing. I'll never really know why she took that punt on me, but it changed my life so much because I've had loads of gigs since. And similarly, Matthew Rice, he sent some really interesting reviewing gigs my way, including reviewing a tattoo festival, which really made me happy. It was so interesting. And also, he got me to review this book about poetry and music of science by Tom McLeish, who also, I was just thinking about it when I was making my talk. It's been a very bad year for my mentors dying. So everyone else watch out. But anyway, he died very young, unfortunately. He was a professor of natural philosophy, combining physics and medieval studies, and it has been a real inspiration for me. When I reviewed his book, he got in touch, and since then, he invited me to do loads of events, and he was the one who got me on the Physics of Life committee, and all of those things just springboard. If there's one lesson I want to kind of portray, is that science is just such a joint effort, and you just never know how one interaction you have is going to have a knock-on influence later on. So again, this is like one of the most enjoyable and fabulous experiences of my career, which has been being involved with Anne Witheridge, who's here, and in this amazing portrait painting project that I could talk about for a long time, and I'm not going to even go there now. But yeah, it's very exciting. Iris Murdoch, of course. I actually went and visited her in the National Portrait Gallery before I came here as a bit of a morale boost. And then these women, I just want to say, I'm sure I've probably missed somebody important, but every single woman on this slide is someone who has done something to boost my career with zero obligation. Like they were not my mentor, they were not in any way officially in charge of me, but somehow, like just because either they saw something or they just wanted to be nice or helpful, have really, like, honestly, I could give a 1-hour talk about each one of these people individually, and don't worry, I won't. But it is my plan that I'm going to profile all these people eventually, because they're just amazing. And I have to say as well that the room is full of my friends, and I actually haven't put my friends on the slide. That doesn't mean I don't love you. It's because I'm talking about my career. So yeah, finally, I just wanted to do a shout out to my family. I'm very lucky to be in a massive family, and yeah, now that we're all adults, it's a really amazing experience. Having so many siblings, especially my sisters, some of whom are here and some of whom are on the live stream. And yeah, and then of course I want to talk about my downwards and across family, my lovely husband Nick, my children Netta and Saul, who I'm trying to get into early scientific experiences, as you can see from some of these pictures here. And then, yeah, this is just my thank you slide. There are like millions of other people who should be on it, but yeah, that's it from me. Thank you.
I get the feeling that's going to be quite hard to follow, actually. So, yeah, I mean, Rivka, she's definitely still standing in our department. She was here right at the start of the restart. She has frequently told me, but I'm not a chemist. And I've been trying to persuade her, actually, you are. I mean, chemistry is molecules, and, you know, it's what molecules do, it's what they make. And there are two things really about Rivka that epitomize what we wanted out of the department and why, you know, the department did so well. And one is that we didn't just want to be what people thought of as chemistry, the chemistry discipline. And in the end, we became renowned for chemistry of life. And the reactions that, you know, at the beginning of Rivka's talk, all those sort of machines of life, it's all chemistry. It's all really complicated, and a lot of chemists don't want to get involved with it. And so, you know, that's biology in action like that, are chemical reactions. But the other thing was, and Rivka didn't actually start out as a chemist. In fact, she still doesn't think she's a chemist. I did start out as a chemist. And I think it's pretty well known, the culture of chemistry and in physical sciences, it's evolved a bit, but it's perhaps not the most inclusive, we could say. There aren't actually a lot of women, still. And one of the things we really wanted to do in the department was to have a different culture, be really positive, be really friendly, be really open, be diverse. And I mean, you can see from Rivka's talk and her approach, because we wanted to be very interdisciplinary. She just epitomizes everything about our department, and she's just been fantastic. Her pastoral care, I have to say, is absolutely exemplary. She was fantastic in so many roles. And one of them was our postgrad, you know, and looking after them and different types of people with all sorts of issues, and she was so helpful. And she's been brilliant, as was mentioned in the faculty, rolling this out and you know, changing the face of doctoral training and looking after people. And she's still doing it. She's another person who says yes quite a lot, although she says no a lot more than Andre. Rivka can be quite firm, I've found. If you catch her at a certain moment, she's like, no, 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 not even. So anyway, if you can join me in thanking Rivka.
I won't keep you too much longer between your seats and the refreshments we've got for you, but I would like to say just a few closing words, because we've really had two wonderful talks and closing comments, which I think reflect really that special sense of collegiality, of working together, of an openness of doing things differently in the chemistry department here at King's. What I really loved about both of our speakers today was the honesty and the humility that they presented with, a career not by design, keeping swimming, and that recognition about the importance of surrounding yourself with great, awesome people who support you, who encourage you, who give you inspiration. I love the Lego, and what I also noticed was you talked about, Andre talked about complexity from the simple building blocks, and Rivka, you were going right down and to the detail, the small things, but what I loved about you, and I suspect your students really value, is how you can take these incredibly complex things and express them so simply. So I think we'll all go away saying, yeah, I've got DNA sorted now, and yeah, this chirality, 2 hands, I've got that sorted. You can express that, and I know your students really value your teaching. But don't underestimate yourselves. You have been promoted to professor. This is a really serious recognition that King's, takes a lot of time and effort and scrutiny. This is a time to be proud. I think on behalf of the faculty, I want to thank you, want to encourage you to continue being amazing inspirations to your colleagues, to your students, and to your family and friends gathered here. So let's have one final round of applause. And do please join us for some refreshments as well.